The 8-box blood typing grid is a simplified surface description masking a staggering biological reality. The International Society of Blood Transfusion currently recognizes 48 distinct blood group systems, built from 397 recognized antigens and driven by 1,971 known genetic alleles. Conventional serological testing relies on agglutination, watching blood cells clump in a test tube. This only reads the lowest resolution surface of the cell. It tells us if a structure reacts to a reagent, ignoring the actual molecular architecture beneath. This binary classification system has been entrenched in medical infrastructure for decades, reducing complex genomic states down to a simple positive or negative checkmark. Treating a label like Rh negative as a single biological identity generates clinical ambiguities. Resolving these discrepancies require abandoning the old grid and moving toward full, long-read genomic sequencing. The ABRH negative label represents a statistical intersection of two independent genomic systems. The label is generated by two systems on different chromosomes, the ABO locus on chromosome 9 and the RH locus on chromosome 1. Operating via different mechanisms, their conjunction in a single person is a mathematical overlap, not a coordinated genetic program. Since these loci are unlinked, two patients bearing the exact same AB negative clinical label may possess fundamentally incompatible Rh genetic architectures right beneath the surface. These two loci also have entirely different evolutionary histories. This timeline shows the AB polymorphism as a deeply conserved trait, maintained by balancing selection over tens of millions of years. This specific glycan program spans multiple non-human primates, predating modern human speciation. The RHD negative deletion represents a much more recent population-specific restructuring of a single protein system. Beyond transfusion rules, ABO glycans modify hemostatic regulation, specifically the clearance of von Willebrand factor and factor VIII in the bloodstream. People with non-O blood groups maintain higher circulating concentrations of these factors. Because the AB phenotype combines both A and B structures, it sits near the high end of the clotting factor distribution. The AB phenotype is a dual glycosylation state. It affects the endothelium, epithelial tissues, and baseline thrombosis risk throughout the body. Meanwhile, the negative side of the label hides even more complexity. This is a 3B structural model of a red blood cell membrane. Even in a standard Rh negative cell, the wider Rh membrane complex, including the RHCE and RHAG structural machinery, remains entirely intact. What actually makes a cell Rh negative depends on a person's demographic history. In European and West Eurasian populations, the typical molecular architecture is a homozygous complete deletion of the RHD gene. The Basque populations of the Iberian Peninsula represent a prime example of this demographic drift, featuring exceptionally high RHD deletion allele frequencies approaching 47%. Outside of Europe, that deletion architecture falls apart. In East Asia, nearly a quarter of serologically Rh-negative Chinese individuals actually retain an intact RHD gene. This is known as the DEL phenotype. Most of these cases are driven by the Asian type DEL mutation, a specific substitution at position 1227. This point mutation produces an extremely low-density D antigen that easily evades standard serological detection. In African diaspora populations, a third architecture emerges. Standard tests often mask complex RHD pseudogenes and RHD-RHCE hybrid genes. Because RHD and RHCE share extreme sequence similarity, they are prone to unequal crossover, exchanging genetic material to form fractured hybrid structures. Conventional short-read sequencing struggles to make sense of these structural variants, leaving approximately 25% of complex Rh cases clinically ambiguous. Transfusing a partial D patient based on a binary serological test carries high clinical risk. Even if their blood is labeled negative, they remain fully capable of forming anti-D alloimmunization against the specific epitopes they lack. The clearest clinical theater where differentiating true RHD negativity matters is maternal-fetal incompatibility during pregnancy. This diagnostic flowchart maps the shift from broad serology toward maternal RHD genotyping and cell-free fetal DNA methods. It branches to differentiate complete RHD deletion from the DEL phenotype. Identifying the Asian-type DEL spares thousands of women from unnecessary anti-D immunoglobulin prophylaxis. The logistical consequence is immense. Redirecting these interventions could increase the effective supply of genuinely needed Rh-negative blood units by roughly 30%. The emerging clinical model is no longer positive versus negative. It is a precise cascade evaluating antigen density, the specific molecular allele, and exact immunization potential. The AB-Rh-negative erythrocyte functions as a direct substrate for engineering universal blood. These cells naturally lack the D antigen, and removing their A and B glycans transforms a niche inventory, normally restricted to AB recipients, into a universal O-negative-like state. Converting these cells maximizes the utility of a rare phenotype that is otherwise operationally limited on the red cell donation side. Researchers use specific enzymes derived from the gut bacterium Akkermansia muciniphila to efficiently shear off extended A and B glycan structures from the cell membrane. While this enzymatic stripping theoretically creates an O-negative mimic, residual immunoreactivity remains a significant clinical obstacle, and as we established earlier with the 48 known blood group systems, merely stripping away A and B glycans is insufficient to achieve true universal compatibility. To move beyond surface-level enzymatic stripping, scientists are using CRISPR-Cas9 to genetically decompose these blood group proteins into precise compatibility modules. Engineered precursor cells tolerate multiple antigen deletions because their residual cytoskeletal architecture, like the band 3 complex, provides compensatory support for the membrane. The ultimate goal is constructing antigen-minimized, genotype-indexed erythrocytes, tailored to solve the logistical nightmare of rare donor transfusions. The dense intersection of ancient glycan programs and population-specific structural variants exposes standard serology's inadequacy. It provides the experimental substrate needed to move from a rigid 8-box taxonomy toward a system of programmable blood.